Have you ever tried designing a gRNA? What are the key factors to consider when creating one? What makes a gRNA truly efficient, specific, and with minimal off-target effects? If you're looking to design the perfect gRNA for your CRISPR experiments, RNABase is here to guide you. In this video, we'll show you how to design a high-efficiency, high-specificity gRNA with low off-target risk, step by step. Let's start with rule number 1, choose the right target region. When using gRNA to interfere with gene expression, where you target matters just as much as what you target. We recommend designing gRNAs in the second or third exon after the start codon, ATG, and here's why. If the gRNA is placed too close to ATG, the cell might still initiate alternative translation, producing a shortened protein that still retains partial function. Conversely, targeting sites too close to the stop codon might result in C-terminal truncations, which can also leave behind a functional protein. To maximize gene disruption, it's best to avoid both ends of the coding region. At RNABase, we default to and recommend targeting the second or third exon after the ATG-containing exon, giving you the best chance of completely silencing your gene of interest. Design rule number 2, use the right gRNA length. Not all gRNAs are created equal, and length matters. Different Cas proteins require guide RNAs of different sizes. Depending on the system you're using, the optimal gRNA length can range from 19 to 30 base pairs. For example, the classic SP-Cas9 system typically uses a 20 BP guide. But newer variants like Cas12a or Cas13 may require longer or shorter guides. Using the wrong length can reduce targeting efficiency, or even prevent binding entirely. That's why RNABase automatically adjusts gRNA length based on the Cas system you select, ensuring your guide is both compatible and effective. Design rule number 3, match the starting nucleotide to the promoter. It may seem like a small detail, but the first base of your gRNA sequence can affect expression levels. Why? Because common RNA polymerase III promoters like U6 and H1 have base preferences. The U6 promoter prefers gRNAs that start with guanine, G. The H1 promoter prefers an initial adenine, A. If the first nucleotide doesn't match the promoter's preference, transcription efficiency can drop, which means lower gRNA expression and reduced CRISPR activity. To ensure strong expression, RNABase automatically checks your starting base and optimizes it according to the promoter you choose. Design rule number 4, keep GC content balanced. The GC content of your gRNA affects both binding stability and secondary structure, and striking the right balance is key. In general, low to moderate GC content is recommended. Too much GC can make the guide form unwanted structures, while too little GC may weaken target binding. That's why RNABase defaults to a GC content range between 35% and 65%. A range shown to support both stability and efficiency. The platform automatically calculates and filters gRNAs to stay within this range, so you don't have to worry about hidden inefficiencies in your design. Design rule number 5, avoid internal repeats. Some sequences may look valid, but hidden repetitive patterns can make them unstable or ineffective. Specifically, we strongly avoid gRNAs with internal repeats, such as 6 or more consecutive G or C bases, 6 or more A or T in a row, any long run of repeated single nucleotides like GGGGGG or TTTTTT. Why does this matter? Repeats like these can form secondary structures, interfere with transcription, or cause the gRNA to misfold, all of which reduce editing efficiency. That's why RNABase automatically filters out any guides containing internal repeats, so the sequences you work with are both clean and reliable. Design rule number 6, eliminate palindromes. In gRNA design, palindromic sequences, those that read the same forward and backward, can be problematic. Why? Because palindromes tend to form hairpins and other secondary structures, which can interfere with proper folding, expression, or binding of the gRNA. To reduce this risk, RNABase automatically removes any gRNA candidates that contain palindromes of 8 bases or longer. By avoiding long palindromic patterns, you get cleaner sequences that are more likely to perform well in CRISPR experiments. Design rule number 7, check for off-target matches with BLAST. Even a perfect-looking gRNA can fail if part of its sequence matches another gene in the genome. To prevent this, we run a BLAST analysis to detect off-target risks at the sequence level. Why the C-terminal? Because this region is critical for Cas binding and cutting. So any crossmatch could lead to unwanted gene editing. At RNABase, we use BLAST to scan the genome and ensure your gRNA is uniquely targeted, so you can edit with confidence and precision. After applying all 7 design rules, from target region and GC content to off-target screening, we're left with only the most promising candidates. The gRNAs that pass this rigorous filtering process are predicted to effectively target your gene of interest, while minimizing the risk of off-target effects. These are the guides RNABase recommends for your experiment. Precise, efficient, and ready to use. Want to see what RNABase delivers? Just click the See Example button in the top right corner. A detailed PDF report will pop up, and here's what you'll find inside. Right at the top, you'll see a concise summary of your selected gene. Gene name, species, ensemble ID, transcript ID, transcript and CDS size, chromosome, and exon count. Everything you need to confirm you're targeting the correct gene. Next is a transcripts table, listing all known isoforms of the gene. You'll see each transcript's ID, name, biotype, and whether it codes for a protein. We'll also highlight the transcript you selected, so you can easily keep track. Here, we summarize the CRISPR enzyme you've chosen. You'll get key info including Cas name, species of origin, PAM pattern and PAM length, recommended gRNA length. Next, we provide a visual overview of which exons are being targeted, clearly marked and aligned with your transcript structure. Then, a summary of the gRNA screening process. How many candidates were filtered out by BLAST? How many passed all quality checks? You'll also get a full gRNA table, showing gRNA sequence, PAM sequence, targeted exon, GC%, whether it's recommended, and even a scramble control sequence for each one. This page gives you the complete final sequence, including U6 promoter, guide RNA, scaffold, and terminator, TTTTTT. Ready to copy-paste directly into your expression vector. Still not sure how to design your own gRNA? No problem, we're here to help. If you're a new customer, your first gRNA design is completely free. Just send us an email at info@RNABase.com. And we'll send you a full, customized design report. No strings attached. And there's more. RNABase also offers design services for shRNA, miRNA, and DNA optimization. To explore all our tools and services, visit us anytime at www.rnabase.com. Let RNABase accelerate your research with precision and confidence.